Hi, so this is the part two for the muscle system video. Like, it's a big thing. So it's hard to cover all of it in one video, so that's why I broke it up into two. But anyways, we're going to jump right into it, starting off not with smooth muscle. We're going to start up with ATP sources that I wrote in a way that hopefully you can see, but in case not, I'll list it off and read it for you. And so you sort of have four main sources for ATP or energy in your body. And it's used in different ways, right? First, we have the cytoplasm of the cells themselves that are doing the movements. Most of the time, this is impossible. The energy stores only last for like 1 to 2 seconds if you're lucky. And the only place it really works consistently for a long time is in your eyes, your ojos, right? Because you're always doing like fast twitch, really quick stuff. And so for anything that requires super quick, instant movement, maybe you're using that. But cytoplasmic stores just don't fit the bill for most physical muscular actions. Now we have the star of the muscular show, the creatine phosphate. And so creatine phosphate, when you're doing any actions like say weightlifting, so it'll be easy to remember, it's like creatine weights. You can do it for 5 to 10 seconds before your muscles run out of energy from those creatine phosphates, right? And outside of weightlifting or maybe a 5-second sprint, it's really not that useful. Now you have your glycolysis. We're starting to delve into the deeper range of possible maneuvers for longer actions, right? So 60 seconds, right? So 400-yard dash, like you're going to be using glycolysis, the sugar in your body. But yeah, it's quick. Yeah, it's effective. No, it doesn't require oxygen like the previous ones mentioned. So that's always nice. But it's just not going to pay the bills if you're going to have to, say, run a marathon. But if you're running a marathon, aerobic respiration is where it's at. It's crazy effective. It does need oxygen, which is like why when you're running, you're like, your body's trying to make up for that energy deficit by using aerobic respiration, right? This is where your mitochondria deserve their crown of kings of the body because they produce a crazy amount of energy. And you can do this for hours, right? Humans used to be predators that would just run deer to the ground because when it comes to sprinting or speed, we're sort of dead last in the animal kingdom. But when it comes to endurance, we're second to virtually none. Except for, I don't know, I'm guessing horses can go longer, maybe some of them, but I don't know. I'm trying to be a doctor, not a biologist, right? Well, doctors are biologists. An animal doctor. Yeah, I suppose that'd work too. And so that's it for our ATP sources. And so you can sort of think of that in line with like different physical activities you do with your skeletal muscle. But I'm going to take a break from the skeletal muscle, and break into smooth muscle, of which there's two kinds, phasic and tonic. So smooth muscle, well, there's really one, but they can behave either as phasic or tonic. Phasic, they contract in like waves, right? They're very energy conserving, they're very efficient, and they sort of move in waves, right? Sort of think of peristalsis, which is like when your muscle contracts in a way where it's like a wave. And this is really popular in, say, your digestive tract, which would make sense, right? Because your stomach is made of smooth muscle. And that's how it gets the food out. It slowly contracts and gets that food out. However, that's only phasic. Tonic is a different beast altogether. Tonic, think frozen or stiff. That's how I like to remember it. Phasic is all about the wave. Tonic's all stiff. So, not to bring potty humor into this, but like if you're like asleep and you're not crapping your pants, it's because of these tonic smooth muscles, right? They contract and they get stuck in the stiff phase, which is useful for your sphincters, right? Because that'd be pretty embarrassing, like waking up with crap in your pants. Like that'd ruin anyone's morning, at least mine. And so your skeletal muscle accomplishes this in different ways. First, I'm going to cover, like, this is your average, like, smooth muscle right here. But we're going to take a broad scope and then work inwards as to how they contract, right? And so broad strokes here first. And so we're looking at our smooth muscle cell. They have these different channels, right? You have your classic voltage gated calcium channel. You have mechanical gated calcium channels. And then you have ligand gated calcium channels. If calcium sounds important, it's because it is. It's fetching everywhere, dog. And so calcium, when you activate, these will flood into the cell. And that's how your smooth muscle gets its extracellular calcium, which is where a lot of it, if not the majority of it, comes from. However, just like skeletal muscle, your smooth muscle does have an SR, a sarcoplasmic reticulum, where there is calcium. And so, bringing back G proteins here, fetching cool. Let's say this is like a special chemical needed to activate your G protein. The G protein activates, say, like the alpha part here. The alpha part will then activate the PLC, the phospholipase C, I think it is, and memory slips, which produces IP3, which is needed to open the ryaninium channel for the sarcoplasmic reticulum. And this is where it starts to really pop off. And so that calcium will be sufficient enough to bind to the 4 sites of the calmodulin, which will then unwrap and enable MLCK. And so MLCK is a very important protein that enables the smooth muscle to process, right? And then we have our MLCP, which requires a ton of calcium, which is why it tends to be stimulated by outside calcium. And so this outside calcium will deactivate this row. This row then inactivates, which then makes it impossible for it to phosphorylate the MLCP, and the MLCP is then able to go because there will be no more phosphate on top of it. And yeah, that's the very basic overview of where calcium can come from. You'll notice there's 2 sources, the sarcoplasmic reticulum and the outside. I'll talk more about the importance of this later, but rule of thumb, if it's phasic, it's probably going to be coming from the inside of the cell because it's lower calcium quantities. But if all of these channels are open too and a lot are swinging in from the outside, then it's probably tonic. All right. Okay, so I'm trying not to erase all of my previous work here. Now we're going to get into the nitty-gritty of how smooth the muscle contracts at all. I'll have to remember to talk about skeletal v smooth here. So I'll write that here. Don't let me forget. Okay. First, we're going to discuss phasic. Right? So let's say this is a smooth muscle, right? And it starts out like this, right? We got our familiar actin and all of that. And so it's going to go something like this, ADP. P. It's phosphorylated, it's living the dream. Calcium starts to rush in, right? And this calcium enables the MLCK and ATP to enter. This provides it. Okay, there we go. This gives it another phosphate to play with, which is cool beans. And then the ADP and one of the phosphates will fall off. I know it's super interesting. Sorry, but it's important that we get this stuff lined up. And so I'm just checking my notes here for a brief second. And then afterwards, wait, I got that. Yeah. Okay. I was just doubting myself for a sec. ATP can then rejoin. And here's the neat part. Checking my notes. Yeah, no. Phosphates and then ATP. Then it can go cut straight through, like via hydrolysis, into this next step. So it doesn't have to repay the calcium, MLCK, and ATP bill. You can see how this would be really energy efficient, right? Because I think this is awesome, right? It saves this energy right here, which is why we use smooth muscle instead of skeletal muscle for really time-consuming energy processes. And we can depart for a bit before I talk about tonic and all that. Skeletal muscle is very quick. All that calcium it needs is only in its sarcoplasmic reticulum. It does require a lot of ATP. It's quick, effective, voluntary, wonderful for fight or flight situations. Whereas you have smooth muscle, which is slow, concise, energy conservative, and it's great for stuff that you always need to do for long periods of time because it doesn't require as much ATP, right? The ability to cut through like this is really important because now it doesn't have to worry about like, oh, am I just a waste of resources? No, it's quick. Well, not quick, but it's efficient, which is what really matters for those long haul processes such as digestion, right? You wouldn't want to spend a third of your energy you got from a McChicken just digesting said McChicken. That's where situations like this, smooth muscle shines. But this is only one half of the equation. This is only phasic. We have to talk about tonic. And remember how I was talking about MLCP and how if there was a bunch of calcium, it would disengage the Rho kinase, which would make it the MLCP able to be active and cause it to be tonic? We're going to talk about it here. Oh, and by the way, is if you wanted to return to this original state, MLCP will play a role here to make this possible. But if it just cuts through, it doesn't really worry about it. Okay. Let's erase this half here. Okay. Because this is where the fun begins, right? And so we're going to have MLCP, this step is critical, play in here, which will kick out the other phosphate. So now, just checking my notes, it's contracted like this, right? And so, important thing to remember, MLCP also changes the shape of this myosin head as it's detached. And so that's like, okay, well, why is that important? Brother, it's shaped in a way where it's stubborn as a mule and it's not going to change at all. Well, it will eventually change back and stuff like that, but it's so persistent and like stuck in that taut position, it's called the latch state. And so will be like this. ATPs will try to disengage it, but won't accept it all that well. So now it's just sort of stuck like that. And I cannot overemphasize how important this is. It's because of this we don't crap our pants when we sleep. It is an awesome system. And yeah, that's the phasic system. And am I missing anything right now? I don't think I am. And so this latch state, which is triggered by the activity of MLCP, is quite essential. Think of it like this. I said it once and I'll say it again. It's really important that we get this. If the calcium is from the sarcoplasmic reticulum, it's phasic because there isn't all that much calcium. But if the calcium is triggered by the extracellular channels and all of that opening up, then it'll probably be tonic, because that's enough calcium to inactivate the Rho kinase to make the, what's the word I'm looking for? MLCP active again. All right, and now we're going to talk about stretch and like Golgi reflexes and stuff like that. So on your muscle fibers, you have these spindles, right? This is a spindle, and you can have two kinds here. You can have the bar, which is like multinucleated. Imagine this is like in a larger muscle. Not to scale, but it'll serve its purpose. And the water balloon. That's a scientific name, by the way. It's not really, but I like to think it is. It makes it more fun. So these guys, these muscle spindles, are in charge of detecting stretch. And they have these nerve fibers that go around them to the vertebrae. Okay, where's my red? I don't have red. We'll just use blue then. Oh, I don't like that. And so the bar, that one, it will mainly detect the stretch as it's still or static. The water balloon will help your body know. This is all about just detecting where your arm is. It will let you know if it's moving, if it's stretching while it's moving, which, you know, like I look away, I know my arm's stretching because I have that water balloon tell me it's moving. Now, as I look away, I know my arm's outstretched because I have my bar to tell me. It's very useful and important. That's why we can do all these crazy stuff with our eyes closed. We have muscle memory. And so it goes to this vertebrae, and I'll draw it perfectly. I know I'm an artist. Right. So it looks like this tooth here. And these nerves will wrap around all that. I don't want to ruin this marker, so I'll only do it a few times. But they wrap around, and they go to the dorsal portion, where they will synapse. Right? And this is really important for reflexes, right? And so I'll get back to this later, but the same is true for the water balloon. It can synapse into the dorsal root of your vertebrae, and it'll inform your body, like, hey, this is what's going on, right? And so now we talk about something really cool and interesting. Reflexes. Reflexes, let's pretend this is you have the biggest fetching legs ever, right? And you're at the doctor's office, and they're going to conduct a reflex test. And so, okay, they hit a hammer to your knee, and your leg kicks up. Why is that so? Well, these muscle spindles will detect the stretch that's applied to the muscle or tendon and all that from the hammer, right? Because they hit that muscle or the tendon to the muscle, and it'll detect that and think, oh boy, we need to resolve this. And so there's a few different ways it can try to relieve this stretch. One, it can activate motor neurons to say you know what muscle I'm talking about. I'm sorry, the word slips. Or, a lot of times it does both. It inhibits, like via an interneuron, which will release IPSPs, or inhibitory synapse chemicals, which will prevent a certain muscle from moving, and it'll inhibit this one at the same time. And this is all just reflex to account for that stretch, right, to balance it out. And so, yeah, that's a very basic overview of your stretch reflex. And yeah, now we're going to finish this off by talking about Golgi tendon like reflex. And so I'm going to use a completely different example for this one.
Remember that vertebrae. That's still important.
But, ooh, did I miss anything? I don't believe I did. Oh, okay. So for the bar, right? Remember the strap bar? That is 2. If I can speak, my apologies. That is 2. Whereas the nerve around the ball, the water balloon, is 1A. And so when you read that on a paper, that's what they're referring to. But let's talk about the Golgi tendon stretch reflex. So you have a guy working out really hard in the gym, right? He's got a massive barbell. He's contracting all the way. See, Golgi tendon organs, as the name suggests, are tendons. And they don't detect stress, or they don't detect stretch, but they do detect tension. And you'll see this in the gym. Just go and watch. Like, it could be a guy's first time. He doesn't know how much weight he can lift. But he'll lift like so much weight and his arm will start shaking. His Golgi tendon will detect that and think, oh boy, we're in over our head. Let's pull the brakes here. And it'll send signals to the vertebrae, which usually synapses through an interneuron, which releases IPSPs towards that agonist muscle, like the bicep in this case, as it's lifting it, and it'll just tell it to turn off, essentially, which is why you see the sudden collapses, right? It's the Golgi tendon. Its main purpose is to make sure your muscles don't overreact and say break the bone it's on, or to tear itself up too much. Whereas the stretch reflex is just sort of accounting for that stretch in the muscle and wants to balance it out. That's a brief overview of Golgi tendons. Very interesting stuff. Big fan of it. Very funny to watch. But I believe that's most of it. Thank you for watching, and please have an excellent day.